Inside every single one of your cells, there's this incredible master blueprint, right? A set of instructions that dictates literally everything. But what happens when there's a typo? A tiny glitch in how those instructions get read, one that can turn a perfectly healthy cell into a cancerous one. Well, today we're going to unpack exactly how that happens.
So that's really the big question, isn't it? Our DNA is supposed to be the instruction manual for keeping us healthy. So how in the world does it sometimes write the instructions for something like cancer? To even begin to answer that, we got to go back to basics and understand how the cell reads those instructions in the first place.
Okay, let's dive right in. We'll start at the very beginning. How does information even flow inside a cell? You might be surprised, it's actually a really elegant system.
What you're looking at here is basically the golden rule of molecular biology. It's often called the central dogma, which sounds complicated, but it's not. Think of it this way. The master blueprint is your DNA. That's the permanent, precious instruction book. Then, the cell makes a temporary copy of just one page. That's the messenger RNA or mRNA. And finally, that RNA copy is used to build a protein, and proteins are the little machines that do all the work in the cell. So it's always DNA to RNA to protein. That's the flow of information. It's fundamental.
Now here's where it gets really wild. Pretty much every cell in your body, whether it's in your skin, your brain, your liver, it has the exact same DNA blueprint. So why isn't a brain cell a skin cell? The difference is which parts of the blueprint they use. A skin cell reads the skin pages, a brain cell reads the brain pages. This whole process of picking and choosing which genes to turn on is called gene expression. And honestly, this concept is the key to understanding almost everything we're going to talk about today.
So if cells are only reading certain pages of the book, how do they actually make a working copy of just that one page? Well, that process has a name, and it's absolutely fundamental. It's called transcription.
Transcription is pretty much exactly what it sounds like. It's about making a copy. The cell creates a temporary RNA copy from that permanent DNA blueprint. The analogy I love for this is a library with a priceless ancient book. You're not allowed to take the book out, right? So what do? You find the one page you need and you make a photocopy. That photocopy is your RNA. It's disposable, it's usable, and the original stays safe.
And here's what that whole photocopying process looks like up close. I know, it looks complex, like this incredible molecular machine, because it is. But the basic idea is really simple. The DNA double helix unwinds just a little bit, and one of those strands is used as a template to build the new RNA molecule. The main thing to remember is the end result. You get a finished, working copy of a gene's instruction, and it's ready to go.
Okay, so who's the star player in this whole operation? It's a powerhouse enzyme called RNA polymerase. This is the little machine that does all the heavy lifting. It literally latches onto the DNA, zips along reading the genetic code, and builds that RNA copy, letter by letter. You can think of it as the ultimate reader and writer all rolled into one.
But wait a second. The human genome is massive, billions of letters long. How does RNA polymerase know where to even begin? It's not just reading randomly. Well, it looks for a very specific landmark, a DNA sequence called a promoter. And a promoter is basically a giant neon flashing "Start Here" sign. It tells the polymerase, hey, this is the spot, bind here and start copying.
The whole thing happens in 3 really straightforward stages. First, you've got initiation. That's the polymerase finding the "Start Here" sign. Then elongation, that's the fun part, where it's actually building the RNA strand. And finally, termination. It hits a stop sign, and the brand new RNA copy is released. Start, build, stop. Simple as that.
All right, so we've got a handle on how a copy gets made. But honestly, the most important part, especially when we're talking about diseases like cancer, is figuring out how all of this is controlled. This brings us to the cell's master control panel.
Think about it. A cell can't just have every single gene turned on all the time. That would be absolute chaos. It'd be like turning on every appliance in your house at once. So instead, it has this incredibly sophisticated control panel to turn genes on and off moment by moment depending on exactly what the cell needs to do.
And what are the switches on this control panel? Well, they're mainly proteins called transcription factors. These are the real gatekeepers. They can bind to the DNA right near that "Start Here" sign, and they make a decision. They can either help RNA polymerase get started, which turns the gene on, or they can get in the way and block it, turning the gene off. They're like the bouncers for your genes. But it's not just about tiny protein switches. Control is also physical. It's about accessibility. Sometimes the DNA is all loosely packed, kind of like an open book.
We call this euchromatin, and it's basically open for business. It's easy for the machinery to get in and read the genes. But other times, the DNA is wound up super tightly and locked away. That's heterochromatin, and it effectively puts a do not disturb sign on those genes.
And believe it or not, there's even another layer of control. The cell can add these little chemical tags, they're called methyl groups, directly onto the DNA itself. This process is called DNA methylation, and you can think of it as a much more permanent off switch. It's like putting a lock on that page of the instruction manual, making it physically impossible to read.
So, we've just mapped out this beautiful, intricate, and incredibly well-controlled system. It's amazing. But now we have to ask the critical question. What happens when it all goes wrong? What happens when these controls fail?
To really get a grip on cancer, you need to know about two key types of genes. First, you have proto-oncogenes. Think of these as the cell's gas pedal. Their job is to promote growth and division. Then you have tumor suppressor genes, and you guessed it, these are the brakes. They tell the cell when to slow down or stop dividing. In a healthy cell, it's all about the balance between the gas and the brakes.
And this is exactly where the system can catastrophically fail. Cancer happens when mutations throw that delicate balance completely out of whack. A mutation can jam the gas pedal to the floor, turning a normal proto-oncogene into a superactive oncogene. Or another mutation can just cut the brake lines entirely. Gas pedal stuck down or brakes that don't work, either way, you get the same terrifying result: uncontrolled runaway cell growth.
This really gets to the absolute heart of the problem. There's a quote from the source material that just nails it. It says that a single mutation in a promoter, remember that start here sign we talked about? That one tiny error can lead directly to the messed up gene expression that we see in cancer cells. It's a tiny change with massive consequences. Okay, I know this can all sound pretty grim, but here's the good news. Understanding exactly how this instruction manual gets corrupted is also our single greatest weapon in the fight against cancer. This knowledge is paving the way for much smarter treatments.
And the basic idea is just brilliant. If we can figure out the exact broken switch, the specific protein that's gone haywire in a person's cancer, we can then design a drug that goes after only that broken part. It's like a sniper instead of a bomb. It targets the problem and leaves healthy cells alone. This is the whole idea behind targeted therapy.
Let me give you a real-world example. Some types of breast cancer are driven by way too many of those gas pedal proteins on the cell surface. A common one is called EGFR. With so many of them, they're constantly screaming at the cell to grow. This, of course, leads to way too much transcription of growth genes. So what did scientists do? They designed drugs that specifically seek out and block just the EGFR protein. It shuts down that relentless growth signal right at the source, and that can slow the cancer's growth. It's a perfect case of finding the broken switch and turning it off.
So, there you have it. We've gone from the fundamental flow of information in a cell to the tiny molecular switches that keep it all in check, and we've seen how just one broken switch can lead to cancer. And as we get better and better at reading this incredibly complex genetic blueprint, it leaves us with a huge, hopeful question for the future. Will we one day be able to move beyond just treating the breakdown and actually go back and fix the typos in the blueprint before they can ever cause a problem?